Okay, class. So this is lecture 7. Um, and this lecture is on the second half of mutation. So the second half of mutation, we're going to talk about cancer. We're going to talk about the results of mutations, and then also we'll follow up with ways to repair things. So when we talk about cancer, cancer is usually comprised of cells that are doing four things. So cancer, generally, we're talking about somatic cells. We're not talking about gametic cells. These are body cells. And so, generally, the first thing that is involved in cancer is what we call tumorigenesis. And tumorigenesis is uncontrolled cell growth. So, hopefully, this is a review, but I'm going to draw the cell cycle up here again. And this is basically a piece of pizza, and we cut it into four sections. So, we have the G1, S, G2, and M phase, and then we have G0, which is the rest, cells at rest. So generally your cells are at rest until they get a signal to come out of rest and start dividing. Cancer cells or normal cells generally have like a neighbor function, so that if you have, like, let's say you get a cut. So these cells here and here realize they have no neighbors, and they're gonna be at rest, but then they're gonna go through the cell cycle, and they're gonna divide until they touch their neighbor, and then fill in that cut or whatever, and now they stop dividing. So this cycle can be turned on or off all the time. In cancer cells, they don't recognize their neighbors. They don't care, and they continue to divide, and then you get a tumor, right? And so that's the first step. So tumorigenesis, uncontrolled cell growth, cell cycle is always on. The second step is metastasis. So metastasis is when cancer cells move, right? So lung cells don't hang out with lung cells anymore. They go to the brain, or you know, breast cancer cells go to the lymphatic system. They're moving, and when they do this, this is a particularly dangerous, because imagine you have lung cancer and then your lung cells are moving into your brain. So that's not necessarily good for your brain, because it's not helping you think. In fact, it's interfering with the functions of the cells in your brain, the neurons, by spreading them out and moving them around. And in essence, you're actually disordering an ordered system. And one of the things I teach in 181 is the one of the most important things for life is order. So we have to have order. If you shoved an ice pick into your head, you're disordering an already ordered system. And so that's what kills you. And in fact, this is how cancer kills you, by disordering normally functioning organs. And that's by moving. And the reason it does this is because there are receptors called cadherins and catenins. And let's just say that you have a molecular area code on your cells. So these are receptors, but I'm just assigning random numbers to it to illustrate a point. And let's say that this 0 gets mutated to a 3. So now, instead of area code 602, you're at area code 623, right? This is Phoenix. This is Glendale. And so the cell thinks it lives somewhere else, so it's going to move. And normally cancers have a very specific way that they metastasize. So, like, normally lung cancer cells will go to the brain next. Testicular cancer cells go to the lungs, and so on. And that's because their area code is very close, right? These chances are that you're not going to have these two mutations, so that they go to a completely different state or whatever. All right, so we have metastasis. The next thing is angiogenesis. So angiogenesis. And that is recruiting blood vessels. Right, so without blood vessels, the tumor, it wouldn't matter where it moved, it still couldn't feed itself and eventually it would die. So we have tumorigenesis, metastasis, angiogenesis. Cancer cells have a way of recruiting blood vessels to feed themselves. And then the last thing is immortality. So cancer cells are immortal. This has to do with telomerase that we talked about previously. Right, I have cells from children that I took skin cell samples from. So we call this fibroblast. And I know that those things are only going to divide about 50 times, and then the ends of their chromosomes are going to get too short, and they're going to undergo apoptosis, which is cell suicide. This isn't true for cancer cells. I have cancer cells called THP1 cells in my lab, and they're, they were harvested from a boy that had leukemia in 1972. And as long as I feed them, they continue to divide and grow. So they don't care. They can go through hundreds of thousands of replications because they're immortal. And this is one of the most important genes to lead to immortality. So you know, if you even if all this happened, if a cancer cell could only divide 50 times, eventually the cancer would die, right? It would undergo apoptosis too, but it doesn't. So all of these mutations have to occur in order to make cancer super dangerous. And when you have uncontrolled cell growth or cell cycle, generally that's because there's nothing inhibiting it. So we have checkpoints in here. This one is to a growth checkpoint. So this is growth or gap one. And so it want to make sure that the cells are big enough and there are enough nutrients in the environment to continue through S phase, which is where the DNA is copied. So we have, like, in humans, we have 23 pairs of chromosomes. We have 46 chromosomes in G1. At the end of S phase, we have 92 chromosomes. And in most extra chromosome diseases, like Down syndrome is really the only survivable uh, trisomy number 21. So they have three number 21 chromosomes. Anything else is generally causes death either before birth or slightly after birth. Most children don't survive any extra chromosomes beyond the age of 2. And there are no survivable examples of having too few chromosomes. So this is a really like a death sentence. You cannot go back and undo the division. So once you go through S phase, you're committed. There are things in here we don't talk much about, but there are checkpoints in here that ensure that the DNA there's not too much DNA is replicated. There's these probably fall in this checkpoint. So there's things like P53 that make sure the DNA is replicated correctly, that check it. RB, there's other things that are checking the DNA, making sure that not too much DNA is being produced, that the DNA that's being copied is okay. Um, and there's lots of things that can interfere with this. So we talked a little bit about laying out in the sun and causing UV damage, right? So helps lead to cancer because it causes mutations in these in the DNA, which leads to either cell death, right? So these checkpoints are going to make sure that the DNA is replicated correctly, and if it's not or it can't be repairable, then it undergoes apoptosis, which is cell suicide. If it's fine, then these proteins direct it to M phase, where it completes the cell cycle and then makes two new daughter cells. Right? So this is cancer is due to generally irregularities in the cell cycle, and we're going to talk about that. Okay, so there's a careful balance between making cells and killing cells, right? And so cells undergo a certain number of replications. They're going to undergo apoptosis. Cells get damaged. They undergo apoptosis because it's better to have a cell undergo apoptosis, kill itself, then to pass on mutations. All right, so I mean, you know this. Hopefully, you learned this in intro bio for majors. New cells are formed through cell divisions, which is called mitosis, and then cells that are damaged, mutated cells or whatever, are removed through apoptosis. So just a reminder, apoptosis um, usually involves a lysosome, and in the lysosome we have all these destructive enzymes called DNases and RNases. Collectively, these are called nucleases. And then we have proteases and lipases and carbohydrases. My handwriting's terrible. So all these things are destructive enzymes. They're designed to break down proteins and lipids and carbohydrates and nucleic acids in the cell. And so if the lysosome is opened, then the cell is going to die. Like all these um, macromolecules are going to get chopped into bits. And so that's what apoptosis is. So when we talk about the cell cycle, right, we have to talk about these checkpoints. A lot of these checkpoints are designed to make sure that there's no problems, because if there is, then the cell undergoes apoptosis. And if there isn't, the cell undergoes M phase or mitosis. Right, so there's two options here. And then here's S, G1, G2, and then G0, which is the rest phase. All right. So cancer results when mutations can cause excessive mitosis or insufficient cell death. So something is messing with the cell cycle. Either it's going too fast, or the checkpoints aren't in place, so there's nothing to prevent this or send the cell to apoptosis. All right. So when we talk about cancer genes, we talk about two kinds of different genes. We talk about proto-oncogenes. So proto means before, oncogene is a gene that enables the cell to progress through the cell cycle. So I kind of think of them as go genes, right? When we have the cell cycle here, their job is to make sure that the cell undergoes cell division. So it turns on the cell cycle or keeps it going. It's a green light gene. And so mutations in this that cause it to become an oncogene, so we don't call it proto-oncogene anymore. It's no longer a proto-oncogene, it's simply an oncogene. So mutations that make it too active, right? So the cell cycle is going too fast. This is speeding it up. We're getting too many cells produced, right? And that leads to tumorigenesis. Now, tumor suppressor genes are genes that do exactly what their name says. Their job is to suppress tumors, right? And so tumors are made out of too many cells. So tumor suppressor genes' job is to be like red light stop genes. I think of them as stop genes. So their job is to stop the cell cycle, hold it up, and then hold it up so that the cells can get enough nutrients or repair any DNA damage that's made or whatever before it goes on to M phase. Because once you start into M phase, you basically have to complete it. There's really no option there. And there are some chemotherapeutic drugs that can arrest the cell cycle in M phase, but these cells, these chromosomes are really tightly packaged, like we talked about before when we were talking about histones. And so they're basically like scrolls that are shut down. So your housekeeping genes, the genes that you need to process glucose or whatever nutrients into ATP or whatever, those genes are turned off too. And so the cell would never survive too long being stuck in M phase. And in fact, that's what a lot of chemotherapeutic drugs do. Their job is to stick the cell in M phase, and then eventually it dies, right? The problem is that these drugs kill indiscriminately. They kill all cells that are going through the cell cycle. Now, not all of your cells are going through the cell cycle, but some of them are. Usually these are the ones that people complain about, like the cells that line your digestive system. So, think about like having a sunburn inside your digestive tract, where those cells are being killed, or skin cells, you get photosensitive, or your hair falls out, or you have like blood issues because your cells are constantly being replaced, your red blood cells, your white blood cells, and so on. So anyway, tumor suppressor genes, they're designed to stop or slow down. So these mutations make genes inactive. They no longer can stop the cell cycle. So either case, we're going to get this cell cycle to continue. All right. So I'm not going to go over this again, but G0 is rest, G1 is gap 1 or growth 1. There's a checkpoint here to make sure that it's big enough. We have the DNA synthesis phase, which is S phase here, G2, which is growth or gap 2. And then this is where we have that DNA mutation checkpoint. The enzymes involved in this are like P53 or RB or lots of other ones. And then, there's two options here. Either we undergo apoptosis or we go on to mitosis. You can't go back. Once you've committed to S phase, you're toast. Either you go through the cell cycle or you kill yourself. Okay, so I'm going to show a slideshow here because these are multiple slides that I layered on top of each other. Let's see. Slideshow from current slide. Okay, so RAS is a proto-oncogene. And so this proto-oncogene job is to say go, right? To continue the cell through the cell cycle. So the RAS protein signals the cell to start dividing. But this only occurs when we get the proper extracellular signal. So a signal on the outside of the cell. So here's our plasma membrane. The RAS protein is coupled to a GDP. So this is G diphosphate, right? Guanine. Very similar to ATP. It's an RNA derivative. It's if it was a deoxy, you would have a D in front of it. That would be DGDP. So I know this is RNA because it doesn't have that. And this is just like your ATP. So it can it can get an extracellular signal. That signal can cause phosphorylation. So in this case, the GDP, we add a phosphate to it. That phosphate converts it into GTP. Remember, the most important thing in biology is shape. So once we add a phosphate, we change its shape. And we talked about this before. You could have a shape that's inactive, add a phosphate, cause that protein to become active. And that's exactly what's going on here. Once it becomes active, it gets transferred through the nuclear pore, through the nuclear membrane, and then it activates DNA transcription that produces RNAs that produce proteins that are involved in the cell cycle, right? Now, if we didn't have an extracellular signal, then we wouldn't have an active form of RAS, and we wouldn't get any RNA to tell the cell cycle to go. So remember we said that a proto-oncogene becomes an oncogene whenever it becomes mutated. So we have a single base substitution, converts RAS into an oncogene. Remember we number these from the amino to the carboxyl end. So this is amino acid number 12, and its glycine is converted into valine, right? And that mutation is shown here. It's actually a mutation that affects the cell cycle. So this is how it does it. This isn't a layered slide, so In this case, um, we don't need the external signal, right? RAS is always active. There's lots of mutations that occur to protein, so it could be active or inactive. In this case, it's just always active. It has that shape to make it active, and so it's always being translocated into the nucleus, where it's always turning on the DNA, which is always making RNA, which is always telling the cell to go through the cell cycle. So in this case, we're going to get uncontrolled cell growth, right? And that leads to a tumor. All right. So RB is the opposite, right? This is a tumor suppressor gene, example of a tumor suppressor gene. This stands for retinoblastoma. It's a childhood disease that's relatively rare, but scientists are now figuring out that this is involved in a lot of cancers. So it prevents the transcription factor, right? Remember we talked about transcription factors. So these are proteins that activate transcription. They tell the RNA polymerase where to go. These are generally found in the promoter region. And then one of them that I showed you before is called E2F. I don't care that you know that. I'm just telling you, this transcription factor is bound up by retinoblastoma most of the time. And so because it's bound, it's not free to interact with DNA and turn on the gene, right? So once this is a cyclin-dependent kinase. So CDK is cyclin-dependent kinase. And kinase, you should remember this. So I think I said this already, but most human diseases are caused by mutations in kinases. So kinase's job is to add a phosphate. This particular kinase here is dependent on cyclin, in this case cyclin A, to make it functional. So without this doesn't function. It can't phosphorylate retinoblastoma, right? And cyclins are named for the way they are through the cell cycle, right? They go up and down. Their level goes up and down. Throughout the cell cycle, depending on what phase you are, G1, S, G2, and so on. And that's why they were named, because they cycle through that's why they're called cyclins. They cycle through the cell cycle. Okay, so anyway, this is the normal process. Cyclin, during the stage, right, during S phase, during synthesis phase, the cyclin dependent kinase, which is always present, interacts with the cyclin, phosphorylates retinoblastoma. That phosphorylation changes the shape of RB and it frees this transcription factor, right? That transcription factor then turns on RNA transcripts, right, that are read into proteins. Enzymes are proteins, and those proteins are involved in DNA synthesis. So in this case, the normal function of RB would be to inhibit DNA synthesis so that we don't have too much DNA produced. So it's kind of a stop, right? It's a stop for DNA synthesis when it's functional. So inactive RB causes retinoblastoma, like that childhood disease that we're talking about. So without RB, the E2F can promote transcription of the DNA synthesis genes all the time. This can't be bound up, right? So the shape of RB always allows this to be expressed, and then you can have. So normally it would stop DNA synthesis, but in this case, it's going to turn it on. So it's going to go because it's going to uncontrollably make these enzymes, regardless of whether or not the cyclin dependent kinase is present. Hopefully that makes sense. Okay, so a cell without a functioning RB gene won't stop, but it will continue to divide. Right? It won't stop at G1. It it's probably closer to S phase, but in any case. So we have our G1 checkpoint. If we don't have anything stopping that, then the cell is always going to go through S phase. And once you go through S phase, you're inevitably going to go through M phase. So in the end, since there's no stopping, no ability to stop in this tumor suppressor gene, we get lots of cells made and we get the same outcome, right? We get a tumor. Okay, so earlier we talked about proofreading mechanisms and that the replication machinery generally only makes mistakes 1 per billion nucleotides. But it's not like it's just typing out a 32 story novel. It's it has a lot of proofreading going on here. And so DNA polymerase 3 has the ability to proofread, and remember it can back up, so it can do that in exonucleolytic activity. We have a mismatch repair that can repair mismatched nucleotides. We have base excision repair where the base is basically excised. So just to remind you, we have the sugar, and this is the phosphate. This is the carbon, 5' carbon. So this is 1, 2, 3, 4, 5. Um, we have hydroxyl hydrogen. This is DNA, and then here's our base. So remember the base, the sugar. This is DNA, deoxyribose, and then the phosphate. And then we have the next base here. So we can replace the entire nucleotide, which is the phosphate, sugar, and base, or we can clip the base off and replace just the base. So base excision repair is just replacing the base on the number 1 carbon. Nucleotide excision repair is replacing the whole nucleotide. Are other repair mechanisms, error-prone repair. We'll talk about this. This is kind of like an SOS repair. And then how we repair double-stranded breaks. Like we talked about, ionizing radiation can cut your chromosome. So how does that get repaired? Okay, so DNA polymerase 1 and polymerase 3 both have exonucleolytic activities, so they can go back and they can replace incorrectly incorporated nucleotides. This is for eukaryotes. Prokaryotes have a higher rate of this because they don't really care about mutation that much because they're making, millions of offspring, that, thousands of offspring. Whereas, in eukaryotes, usually it's only a few offspring. So mistakes are much more costly because if you have 100,000 kids, only one of them needs to survive. So you could have a lot more errors in that. As opposed to eukaryotes that have to be more concerned about being more precise when they're doing replication. So polymerase inserts the wrong nucleotide in eukaryotes, about 1 in every 100,000 nucleotides. If you looked at some eukaryotes, that number is probably 1 in 1,000 to 1 in 10,000. Normally, the polymerase can detect the error, so we're talking about polymerase 3 in this case, and it removes the nucleotide and then replaces it with the correct nucleotide. This proofreading mechanism reduces the error rate to about 1 in 10 million, not quite the 1 in 1 billion that I had told you, but there are other mechanisms besides just looking at the incorporated nucleotides to fix things that the polymerase misses. So we have mismatch repair. There are enzymes that go around proofing the DNA for mismatches. In this case, it's showing that a G has been incorrectly paired with a T. So remember that we have the nucleotides that have different shapes, and so that's going to create bulges um, in the double helix. So here we have a slight bulge from the mismatch. A mismatch repair protein comes along, it detects that mismatch. It would be kind of like you running your hand over your skin and then you feel a bump. And so it's going to cut out that mistake and then repair it. Now, one of the ways that so there it could be this strand or this strand, right? We don't know which strand it is. So one of the ways that it can distinguish between the two strands, and then this is an E. coli. So there's an enzyme called methylase, and it adds methyl groups, in this case here. So normally there would be a hydrogen, but it's methylated to the A bases, right? So this is an adenine. Um, and so here this is just showing that this strand is methylated, this strand is methylated, right? So the original strands are methylated. This is representing a fork, right? And the direction of replication is in this direction. So mismatches on the newly synthesized strand. This is the so it has to copy 5 to 3. So this has to be a 3' hydroxyl. So this is the leading strand. This is the lagging strand. So we have a mismatch here on the lagging strand. This strand is methylated, so it knows that it needs to replace the nucleotide on the non-methylated strand to make sure that the mutation follow the mutation is not carried on, right? It uses the original strand as the template, assuming that that nucleotide is the correct nucleotide. So it's going to cut out a section of the newly synthesized DNA, and then it's going to replace it, right? So we're going to get rid of this T, and it will correctly place it with a C. So this red area is showing you the newly repaired region of DNA. And that's called mismatch repair. So that reduces replication error to about 1 in a billion base pairs, which is what the number that I gave you earlier when we were talking about DNA replication. Okay, so we can also just cut out the bases like I talked about. So depurination, of course, a purine is a double ring, right? And that's A's and G's, so you need to know that word because it keeps coming up over and over again. We talked about alkylation, right? So we can have, you know, different things that cause this so that it's mismatched. And we talked about that in a mutation one lecture. So these are non-bulky damaged bases, and they can be recognized. So they're recognized by a glycosylase. It cuts out the damaged base. So it's just cutting the base. It's leaving the sugar and the phosphate group intact. And so that base is removed. So it can either be a pyrimidine or a purine that's excised. And then an endonuclease will cut the damaged strand. So it's showing you here, it's cutting that strand because it knows there's no base there. So a phosphodiesterase, remember the DNA backbone is phosphodiester bonds. Hopefully that's review. So it's going to remove that stretch of DNA, right? It's shown here where the base is moved is removed. The polymerase fills in the gap. Remember, this is polymerase 1. We talked about this before. The same polymerase that removes the nucleotides and does the excision repair. And then DNA ligase. Remember, DNA ligase has to join the gap because no polymerase can do that. So those are the enzymes involved in it. Bulky base mutations can't be fixed by base excision because the bases are linked together. Like these two T's are covalently linked to one another, and so they you can't just cut off a base. You have to cut off this whole section. So here, nucleotide excision repair is it recognizes the bulge due to the thymine dimer. There are lots of enzymes in play. I don't care that you know all of these, but I want you to know that it's a complicated system, complex system. And we talked about this already. So xeroderma pigmentosum is an inherited illness, right, that causes defects in nucleotide excision repair. That means that individuals affected by this genetic mutation, right, this is not due to, I mean, it is due to exposure to UV light, but in a normal person, they would be able to repair those thymine dimers. In a person with xeroderma pigmentosum, they don't they don't have that excision repair, nucleotide excision repair, um, or yeah, to correct this. So they're super sensitive to UV light. They're prone to developing cancer, right? Because remember, exposure to UV light, the most common thing that occurs is cancer, right? Because you're inducing mutations into your system. And if you accumulate too many mutations, you could end up with mutations in either proto-oncogenes or tumor suppressor genes. Okay, so another system is an error-prone repair system. So in E. coli, they again they don't care. They were going to have lots of offspring. So what if a few of them get like a whole ton of mutations? So in E. coli, there's a specific polymerase that can initiate an SOS repair, right? SOS, save our ship, or whatever other word you want to put in there. So these are full of errors, right? The ones that we've already talked about are generally error free and they reduce the mutation rate. But sometimes damage is so bad that making repairs is impossible. Like if you damage both strands of DNA. And so E. coli will initiate this SOS system that introduces mutations, but in many instances prevents death, right? So you have all this mutated DNA. So let's say you have an A mismatched with a G. You don't know what it is, so you're going to invoke this system. You're going to make tons of cells, right? It's not like us where these cells were multicellular and the mutation in one cell is going to affect the other. These are independent, single-celled organisms. So, you're going to you invoke a polymerase that's just going to throw random bases in there. So it might throw in a G and have an offspring with that. So this might be that, and then it would die. Or it may throw in a T here, and then this one might die. Or it could throw in a C, right, and this one would live. Or maybe it throws in, let's see, we covered G, T, C. I guess that's really the only option except A. And then it could do the other strand too. It could do both strands. So there's like 16 different combinations of pairings that you could get, and maybe only one lives, and the other 15 die. Well, it doesn't care because it just wants to survive. And then you can think of this as like being in lots of places in the DNA. So maybe you have a million. Maybe you produce millions of cells, and then only like one or two actually survive. Well, that's okay. You're going to live to fight another day. So this is how it works. There's a clamp that holds this polymerase in place, and that clamp uh, allows progression of the polymerase 3, right? let's say that we have DNA damage here. Well, the polymerase can't proceed through there. It's too messed up and so it causes it to stop. Well, when that happens, the clamp releases the polymerase 3 and adds on a different polymerase called polymerase 5, which is super error prone because it doesn't have the right shape. It's kind of loose on the double helix. And so it can slide past this, but it also isn't very good at reading because it's too far away. Imagine if you had a book and you're doing a typo and uh, you can't really see the letters, you're just randomly putting in letters. That's kind of what's going on here. And so sometimes it gets it right, sometimes it gets it wrong. And then, like I said, maybe a million times it gets it wrong and all those offspring die. But you just need one cell to survive if you're a bacteria to continue on your line. This is not a good system for eukaryotes because it would be super, uh, it would contribute to cancer quite a bit, and to death and mutation. Now, scientists can use this, right? Because sometimes we want to introduce mutations as geneticists. And so we can use these various polymerases that are terrible at proofreading to induce mutations in DNA. All right, so it's only going to go add a few nucleotides, and it's like I said, it's not very good at sitting on the the double helix at all, and so that is eventually replaced, and then the polymerase 3 gets back on and it continues replication. So it's trying to process through a few hundred nucleotides. There's 10 nucleotides to every turn, so this, and the artist is representing, is about 50 base pairs before it switches back. And so the hope is that it's just going to randomly throw in the right nucleotide. Now, we also talked about like ionizing radiation can cause, like X-rays can cause double-stranded breaks in DNA, and highly reactive oxygen species that are byproducts of metabolism can also cause this, right? So your own natural metabolic process and like the electron transport chain, those electrons can fly off of there and produce free radicals. And those, in fact, one of the things in the electron transport chain called coenzyme Q10, they sell this as an antioxidant. Its job is to bind up reactive oxygen species, but it's actually part of the electron transport chain. So if you're at the store, check it out. You'll probably find a bottle of this that they sell to inhibit reactive oxygen species, which it does a very effective job of reducing DNA damage. So as you get older, you might think about using these kind of supplements that bind up reactive oxygen species like vitamin E and coenzyme Q10 and so on, because it helps prevent or repair DNA damage. Okay, so unrepaired breaks can cause major chromosomal abnormalities, and we're gonna have there's a whole lecture on this, and of course that's going to lead to cancer, death, or mutation. So that's not good. Now, I don't care if you know the enzymes. I'm just showing you the enzymes here. So we're Ku80, Ku70, they bind to the ends, right? And then they're joining them together through ligation process. This is non-homologous. There's no way for it to know what direction it can be repaired. So let's say we have a cleave here, and let's just say that this is the telomere, right? So it could, if you had multiple breaks, let's see, multiple breaks like this. So it could insert this in this direction, which would be the correct direction, or it could flip it around so that this strand ends up on the top and this ends up on the bottom. And so basically this is going to be flip-flopped, and it's going to, so this is an inversion. We'll talk about how chromosomes line up, and this happens. So sometimes it makes the correct repairs, sometimes it doesn't. It really doesn't have a guide at all to fix this. And some base pairs are lost when the ends get trimmed, so it's going to have to cut these ends down and then link them together. So we can do error. This is error prone, right? So you can do error free repair, but the only way to do this is we have a homolog. So remember we have homologous or sister chromatids, and let's say we have a double stranded break while the cell's going through the cell cycle. Well, it can use the homologous chromosome or the sister chromatid as a guide to repair it. And this is how CRISPR works as well. So this is a gene editing system. And basically what it does is it targets a certain region, cuts out that DNA, and you provide a guide DNA which acts like this uh, homologous chromosome or sister chromatid. And so let's say that you have a sickle cell anemia mutation. Let's just say it's so. Let's just say that this is 5. I'll make it 3. So I'll make it the coding strand. So we have 3 prime, 5 prime. I'm sorry, this isn't the coding strand. This is the non-coding strand. 3 prime to 5 prime, so the RNA is going to be 5 to 3. And let's say that you have a mutation. So normally this would be a G, but it's mutated into an A, right? And that mutation causes your RNA to be made, and then the proper protein product isn't made. So you want to repair that. So you could make a guide DNA, have CRISPR-Cas9 go in, locate this with homologous, looking at homologous. So there's another RNA that you have to put into the CRISPR-Cas9. It looks for a homologous region, cleaves that out, cleaves the whole thing out, right? So it's kind of like this, like a double-stranded break. It doesn't know what to put in there, so the only thing it can do is use a guide DNA, right? A homologous piece of DNA, like this sister chromatid, to guide it into repair. And so in this guide DNA, you put whatever nucleotide you want it to incorporate. So let's say that you, I drew this, I crossed this out, but let's say that I said that that was a C. So you would have a G in here so that whenever the repair mechanism came in, it would actually incorporate the C back into the original strand of DNA. Okay, so here's the double-stranded break. We have the same end trimming. So enzymes can create what this is called a D loop or displacement loop. And so basically what they're using is the sister chromatid strand as a template to synthesize the new strand. And so the idea is that these strands are identical to one another. Um, and then again, this is going to use the sister chromatid strand as well. So in the end, because it's using its exact copy strand as a guide, then there shouldn't be any mispairings made or any mutations introduced like it would be here. So I don't know why that's transferring over to the next slide, but let me erase this. Oh, maybe I can't. All right, whatever. I'm just going to go on. So here's the D loop, the displacement loop, and we have this strand invading using this strand as a template, and then we get replication there, which is shown here in green. And then, nothing can do that, so you have to have a ligase to seal these backbones here. But this is a much better repair mechanism than the non-specific strains. And I think this must be the last slide. It is. So that's the end of this lecture, which let me just check this. I think this is. Yeah, Mutation 2 is lecture 6. And I think this is my 8th recording, so we would call this lesson 8. Anyway, uh, that's it for now, and I will see you next time.